everybody, I'm Shruti, your mentor at What Next Education, and today I'm going to be explaining membrane structure and transport. Now this is a very core concept that applies to your CSIR exam, your DBT exam and any other entrance exam you're going to write or any interview you go to. These are core concepts that you should know and must master. Okay, now when we talk about the membrane structure, we all know that a cell has a cell membrane, okay, and this cell membrane is actually composed of a lipid bilayer. Okay, and what does that mean? There are two layers. Okay, so fundamentally we have a head and a tail for every lipid molecule. The head is going to be hydrophilic, means it loves water. Okay, but the tail is hydrophobic because it hates water, okay, and it's scared of water. So now if it is scared of water, I can't just put one layer and say, I don't care, you face the water, right? So instead what nature has done is there is two layers like this, okay, wherein the hydrophobic tails are confined on the inside segments and the heads are facing the outside. So this way what happens is it will assemble itself in such a way that the tail is not exposed to the aqueous environment. Okay, now this particular lipid model is called as a fluid mosaic model because these lipids and proteins can move laterally. So the lipids form this bilayer and proteins can move laterally. Now this compound called as cholesterol, okay, I know it is not foreign to you, but this compound is the one that adds fluidity to the membrane. Okay, super simple concept. Now over and above, how the protein is aligned in this lipid bilayer is super important, okay, because in this bilayer we have some proteins that are integrated inside the layer. Okay, now I'm putting two proteins close to each other, but actually in the membrane you have these proteins scattered. So these ones integrated inside are called as the integral proteins. Okay, and these are going to be the ones embedded inside, and these are the ones who can move along. Okay, now we have other kind of proteins that is attached to the outside or through another molecule. Now these proteins make my peripheral proteins. So clearly periphery on the surface, okay, and these are loosely attached, okay, and sometimes these peripheral proteins can bind with another glucose molecule forming a glycolipid, or it can mix with another phosphate molecule and form a phospholipid and so on. Okay, so these peripheral proteins and integral proteins are associated with this bilayer, and this movement of the protein is what gives a dynamic arrangement to the membrane. Clear? But there are certain components like the cytoskeleton that will restrict this protein running away everywhere, or protein-protein interactions can constrict this movement, and as a result, and also lipid rafts are there which can restrict the movement of these proteins. Clear? Now looking at the transport mechanism across the membrane. Firstly, we have diffusion. Okay, so diffusion is the simplest type of transport wherein from a concentration higher region of concentration to a lower region is just going to migrate simply through the membrane. Okay, there is nothing else to worry about. But when you look at osmosis, it is similar to diffusion, okay, but this is where students get a lot of confusion. So osmosis is where there is a semi-permeable membrane. Diffusion is where everybody on this side will happily move on the other side, okay. But in case of osmosis, that does not happen. You have passive movement wherein across a semi-permeable membrane, I have only selective molecules that can go. Everybody cannot happily pass through, only selective molecules will move. Okay, now this is again through a concentration gradient, okay, but here we have a solute and we have a solvent, right? So this concentration gradient pertains to the solute majorly. In case of diffusion, whoever is here higher will move that side. That's all. Okay, it's very simple. But here the water is going to again have some solutes. Okay, so water moves from a region of high okay, so what happens is when water is there, water is basically the solvent. Now when there is less solute, then water will move from a region of higher concentration to lower concentrations. That means to a region where solute is more. right? Now if the solute is going to move, it's not so easy for the solute to move, so it's mostly water that's going to move. So what I want you to remember is there is selective movement of these molecules, and it's mostly water that will go from a region of its higher concentration to a region of its lower concentration. Now why I'm repeating this so many times is because a solute may be high in one end, but it's not going to move. It's the water that is going to move. So that is why I said it is selective. So both solute, water, everybody cannot happily pass. It's only selected molecules like water who is allowed to pass through that membrane. Okay, I hope that is clear to you. Now because of this gradient and because of this particular interaction, there is certain pressure that has to be maintained so that in both levels the osmotic pressure, okay, has to be equalized, so that it's not like they're moving all the time. They have to move and they have to stop at one point, right? So that pressure developed is what we call as an osmotic pressure. Clear? Then apart from that, what else I can do? How else I can transport? I can use ion channels. Okay, and how I will use ion channels exactly? So I will use certain different kinds of channels. For example, I have voltage gated channels. So what the voltage gated channels will do, when the voltage will reach a certain threshold or when there is a membrane potential that's changing, these ion channels will open. So when this channel is open, there is a selective ion flow that happens. Okay, now apart from voltage gated, I also have ligand gated. So when I see certain kind of ligands, I will open my channel and I will allow certain type of ions to pass through. I also have some mechanosensitive ion channels. Okay, now what do I mean by mechanosensitive? When there is mechanical stress, I will immediately open. Okay, now these kind of ion channels are the one who is most important for impulse conduction in our nervous system as well as for muscle contraction in all our muscular tissues. Clear? Now apart from this, next type is our active transport. Now so far whatever I said is very basic. They can just happen on their own. Okay, I don't have to pay money and all. But here active transport is where I have to pay. So what do I pay in a cellular system? I am going to pay as energy. So they require energy and they move against the concentration gradient. Okay. So that is super important to remember that this is the only type of transport that will involve the usage of ATP. Alright. Now here what examples we have is definitely sodium, potassium, ATPase pump. Okay. So here in any system there is an electrochemical gradient. So in case of our axons, right, in our neurons, this is the main pump that is going to maintain sodium potassium balance, and that is what actually helps in conduction of our messages. Okay, so it's super important. Apart from that, we have calcium pump. Okay, very common in our systems. Okay, so they regulate the intercellular calcium levels in the system. Okay, now apart from this, we also have some membrane pumps. Okay, now what these membrane pumps do, they will selectively transport certain proteins. Okay, now membrane pumps are a type of active transport only where they will utilize the ATP. Okay, but here it can be directly using the ATP or indirectly, some gradient is established because of this primary kind of active transport, and because of that gradient I am going to utilize that energy and I am going to create a separate transport out of that. Okay, so I can name this as primary active transport pumps, or secondary. So secondary is because of the gradient that is created due to this primary transport, but primary transport will directly use the ATP. Clear to you? Right? Now in this particular type of membrane pumps or active transport, there is two kinds of systems that we can use. One is a symport, the other one is an antiport. Okay, so port, transport, okay, transporting the molecules. Now symport, same, okay, symport is same direction, both molecules are going to go. Antiport is I have one molecule coming in, one molecule going out. Okay, so opposite. So anti is opposite, right? Very simple to understand in here. Okay, now when you look at a cell, transport is not just moving ions or moving proteins, correct? We have mechanisms to sort and regulate this kind of an intracellular transport. Okay, so if you look at mechanism of sorting and transporting, intracellular transport, intracellular, not inter, inside the cell itself, how it goes. So firstly we have vesicular trafficking. Okay, so what does that mean? Some kind of signals are going to come. Okay, now when these signals come, okay, now there are certain proteins that are tagged with these signals. So when these signals are going to come, these certain proteins are going to be targeted, and once these certain proteins are activated, what happens? There are some vesicular buds that is formed. Okay, now they fuse with the target. So these vesicles are going to contain some signals. So this is going to go to the target. These vesicles are going to go to the target, and they are going to, give in the molecules, and they're going to interact. Now we have extremely specific proteins called the snare proteins. Okay, so these snare proteins are the one that will mediate this vesicle coming in, okay, docking, fusing, all of this, the snare proteins are going to take care. Okay, so what exactly happens? There's going to be a vesicle containing some proteins. Now once the signal will come, these vesicles will go to my target, dock there, fuse with the membrane, release all the contents into the target place wherever I have to go. Okay, now how are these targeting and sorting regulated? Definitely through certain mechanisms like phosphorylation. Okay, phosphorylation or GTPases. All this can help in regulation of my sorting and my transport inside the cell. Clear, everybody? Now over and above this, you have to remember that all the membranes that we are talking about definitely have electrical properties. Okay, everything in our body is the nervous system, right? Without signals and impulses from the nervous system, there is nothing that anybody can do. So there is a certain membrane potential that exists. Okay, and this membrane potential is nothing but a voltage difference across the membrane. Now normally when you are resting, there is a certain potential, and when there is an impulse, there is a certain potential. So that is what we call as resting potential and the action potential. Okay, now when we are talking about the animal physiology part of it, you will understand details of how this particular potential is changing, and how exactly it opens the channels, and how depolarization, repolarization is going to happen, and that is what basically excites every single cell in our body. Okay, so all of these concepts that I taught you today basically comes under this cell biology unit, and trust me, these concepts are asked in every single exam, and even if you're sitting for an interview, they will expect you to understand these basic membrane dynamics, potential, okay, how the transports are happening, and other things in the chapter. Okay, so if you are looking out for hardcore materials, okay, so not just this, in detail explanation from scratch from standard author books, then we have exclusive materials waiting for you, and also we have specialized AI tools that can help in optimizing your self-study preparation and giving you the best practice possible according to your level and your pace of learning. So if you are interested, definitely do visit our website whatnexteducation.com, or reach out to me for a one-on-one call on my Instagram profile, What Next Education. Okay, so it was a pleasure meeting you all today, and I hope this particular concept is super easy and understandable for you right now. So if you have any questions, do comment, otherwise comment a star so that I know that you are ready and you are in the verge of your preparation. Okay, so all the very best, guys, and I'm looking forward to see you all in another class. Take care, bye bye.